The animal cell. All the tissues and organs of the body originate from a microscopic structure, the fertilized ovum, which consists of a soft jelly-like material enclosed in a membrane and containing a vessel or small spherical body inside which are one or more denser spots. This may be regarded as a complete cell. All the solid tissues consist largely of cells essentially similar to it in nature but differing in external form. In the higher organism, a cell may be defined as a nucleated mass of protoplasm of microscopic size. Its two essentials, therefore, are a soft jelly-like material similar to that found in the ovum and usually styled cytoplasm, and a small spherical body embedded in it and termed a nucleus. Some of the unicellular protozoa contain no nuclei but granular particles which, like true nuclei, stain with basic dyes. The other constituents of the ovum, viz., its limiting membrane and the denser spots contained in the nucleus, called the nucleolus, are not essential to the type cell, and in fact many cells exist without them. Cytoplasm, protoplasm, is a material probably of variable constitution during life, but yielding on its disintegration bodies, chiefly of proteid nature, lecithin and cholesterol, are constantly found in it, as well as in organic salts, chief among which are the phosphates and chlorides of potassium, sodium and calcium. It is of a semi-fluid, viscid consistence and probably colloidal in nature. The living cytoplasm appears to consist of a homogeneous and structureless ground substance in which are embedded granules of various types. The mitochondria are the most constant type of granule and vary in form from granules to rods and threads. Their function is unknown. Some of the granules are proteid in nature and probably essential constituents. Others are fat, glycogen, or pigment granules and are regarded as adventitious material taken in from without and hence are styled cell inclusions or paraplasm. When, however, cells have been fixed by reagents, a fibrillar or granular appearance can often be made out under a high power of microscope. The fibrils are usually arranged in a network or reticulum, to which the term spongioplasm is applied, the clear substance in the meshes being termed hyaloplasm. The size and the shape of the meshes of spongioplasm vary in different cells and in different parts of the same cell. The relative amounts of spongioplasm and hyaloplasm also vary in different cells, the latter preponderating in the young cell and the former increasing at the expense of hyaloplasm as the cell grows. Such appearances in fixed cells are no indication whatsoever of the existence of similar structures in the living. Although there must have been something in the living cell to give rise to the fixed structures, the peripheral layer of a cell is in all cases modified, either by the formation of a definite cell membrane, as in the ovum, or more frequently in the case of animal cells, by a transformation, probably chemical in nature, which is only recognizable by the fact that the surface of the cell behaves as a semi-permeable membrane. Nucleus. The nucleus is a minute body embedded in the protoplasm and usually of a spherical or oval form. Its size having little relation to that of the cell, it is surrounded by a well-defined wall, the nuclear membrane. This encloses the nuclear substance, nuclear matrix, which is composed of a homogeneous material in which is usually embedded one or two nuclei. In fixed cell, the nucleus seems to consist of a clear substance or chiroplasm and a network or chirometome. The former is probably of the same nature as the hyaloplasm of the cell, but the latter, which forms also the wall of the nucleus, differs from the spongioplasm of the cell substance. It consists of a fiber or filaments arranged in a reticular manner. These filaments are composed of a homogeneous material known as linen, which stains with acid dyes and contains embedded in its substance particles which have a strong affinity for basic dyes. These basophil granules have been named chromatin or basic chromatin and owe their staining properties to the presence of nucleic acid. Within the nuclear matrix are one or more highly refracting bodies termed nucleoli connected with the nuclear membrane by the nuclear filaments. They are regarded as being of two kinds. Some are mere local condensations, net knots of the chromatin. These are irregular in shape and are termed pseudonuclei. Others are distinct body differing from the pseudonuclei both in nature and chemical composition. They may be termed true nuclei and are usually found in resting cells. The true nuclei are oxophyll, that is, they stain with acid dyes. Most living cells contain, in addition to their protoplasm and nucleus, a small particle which usually lies near the nucleus and is termed the centrosome. In the middle of the centrosome is a minor body called the centriole, and surrounding this is a clear spherical mass known as centrosphere. The protoplasm surrounding the centrosphere is frequently arranged in radiating fibrillar rows of granules, forming what is termed the attraction sphere. Reproduction of cells. Reproduction of cells is affected either by direct or by indirect division. In reproduction by direct division, the nucleus becomes constricted in its center, assuming an hourglass shape, and then divides into two. This is followed by a cleavage or division of the whole protoplasm mass of the cell, and thus two daughter cells are formed, each containing a nucleus. These daughter cells are at first smaller than the original mother cell, but they grow, and the process may be repeated in them, so that the multiplication may take place rapidly. Indirect division or karomitosis has been observed in all the tissues: generative cells, epithelial tissue, connective tissue, muscular tissue, and nerve tissue. It is possible that cell division may always take place by indirect method. The process of indirect cell is characterized by a series of complex changes in the nucleus, leading to its subdivision. This is followed by cleavage of the cell protoplasm, starting with the nucleus in the quiescent or resting stage. These changes may be briefly grouped under the four following phases. Number 1. Prophase. The nuclear network of chromatin filaments assume the form of a twisted skein or spirum. While the nuclear membrane and nucleolus disappear, the convoluted skein of a chromatin divides into a definite number of V-shaped segments or chromosomes. The number of chromosomes vary in different animals, but is constant for all the cells in an animal of any given species. In man, the number is given by Fleming and Duisberg as 24. Coincidentally with or preceding these changes, the centriole, which usually lies by the side of the nucleus, undergoes subdivision, and the two resulting centrioles, each surrounded by a centrosphere, are seen to be connected by a spindle of delicate aromatic fibers, the aromatic spindle. The centrioles move away from each other, one toward either extremity of the nucleus, and the fibrils of aromatic spindle are correspondingly lengthened. A line encircling the spindle midway between its extremities or poles is named the equator, and around this the V-shaped chromosomes arrange themselves in the form of a star, thus constituting the mother star or monastar. Number 2. Metaphase. Each V-shaped chromosome now undergoes longitudinal cleavage into two equal parts, or daughter chromosomes, the cleavage commencing at the apex of the V and extending along its divergent limbs. Number 3. Anaphase. The daughter chromosomes, thus separated, travel in opposite directions along the fibrils of the aromatic spindle toward the centrioles, around which they group themselves, and thus two star-like figures are formed, one at the either pole of the aromatic spindle. This constitutes the diaster. The daughter chromosomes now arrange themselves into skein or spirum and eventually form the network of chromatin, which is characteristic of the resting nucleus. Number 4. Telophase. The cell protoplasm begins to appear constricted around the equator of the aromatic spindle, where double rows of the granules are also sometimes seen. The constriction deepens, and the original cell gradually becomes divided into two new cells, each with its own nucleus and centrosome, which assume the ordinary position occupied by such structures in the resting stage. The nuclear membrane and nucleolus are also differentiated during this phase.